over time. And during hypoxia, what you can appreciate is that this number of autophagisms is considerably more, and they come and they disappear, which means the autophagisms are formed and then they disappear, which means the autophagisms form, then they fuse with the lysosomes and then they disappear. So this clearly suggests that there is increased autophagy during hypoxia. So what I was speaking so far here is a general autophagy where there is no specificity. Anything that is present in the cytoplasm can be targeted into autophagisms for degradation. However, recently what has been known is that there are organelle-specific autophagy. For example, specifically the damaged mitochondria can be targeted into autophagisms, which we call it as mitophagy. And here the nuclear content, the RNA, each and every one of them can be specifically targeted into autophagisms, which requires specific receptors that bind to the organelle or the thing that has to be targeted into autophagisms, and that signal goes and talks to the LC32 normally, and that helps in the targeting of these proteins into the autophagisms where they would be degraded. Since we are speaking about endoplasmic reticulum, this is from one of our collaborators' lab. They had shown that specifically endoplasmic reticulum, the damaged endoplasmic reticulum, can be targeted into autophagisms for degradation, which means that the damaged endoplasmic, there is a process or a specific mechanism whereby the damaged part of the endoplasmic reticulum can be taken into autophagisms and degraded in the autophagisms. And the process works like this. So, as you can appreciate, in this schematic, which was made by Sandhya, we can see that the endoplasmic reticulum here, and there are specific receptors here. These are specific receptors which have been studied by different people to act as ER-phagy receptors or receptors that can target damaged part of the endoplasmic reticulum into autophagisms. And these different receptors, one of the commonalities between all these receptors is this Lear domain, which we call it as LC3 interacting region. And here we have, in the autophagisms, we have the LC3, and these receptors have this LC3 interacting region. Therefore, these receptors can go and bind to the LC3 within the autophagisms. Thereby, the part that needs to be degraded can be targeted into autophagisms. So once it is targeted, then the autophagosome is formed, and as I explained previously, they fuse with the lysosomes, and this gets degraded. So we did look at all these different receptors, but one of the before that, I was jumping a bit. So what we see here is like when we transfix. So what Sandhya did here is she used a plasmid that specifically stains for the endoplasmic reticulum, and the GFP-WIPI that specifically stains for the autophagisms. When she co-transfected these two things, this is a normal cell. And as you see, when the hypoxic, the time, the induction of hypoxia increases, what you see is that for the green and the red signal, they colocalize, which means the endoplasmic reticulum is localizing with the autophagisms. And over time, what you can appreciate here is that this is reducing. Initially, there is a lot that is happening, but then it reduces. So, which means the reticulum fuses with, or the damaged part of the ER fuses with the autophagosomes, but they recover from that once the stress is relieved. And autophagosomes which are formed in a normoxic condition, but you don't really see so many. But in this case, if you see here, during I think you would appreciate that there is a lot of these ER and autophagosomes which are coming together. They colocalize, they come together. suggesting that the damaged there is more damage to the endoplasmic reticulum, but at the same time, the backup mechanism that helps in clearing off this damaged ER is also increased. So as I said, there are several adaptive proteins, and we looked at all the adaptive proteins, but one particular adaptive protein, which is FAM134B, which is a specific ER-Phagy protein. What you can see here is during hypoxia, you would appreciate that the level of the FAM134B increases. But you block the lysosomes using a drug, conchynomycin A, you can see that the levels are restored, which means that during hypoxia, this FAM is taken into autophagosomes and they are degraded. However, when you block the lysosomal activity, the degradation is prevented, therefore you have. So this suggests that possibly during hypoxia, there is this ER-Phagy mechanism that is happening. And when we do microscopy, Sandhya again clearly showed that the FAM134B and the LC3B, which is a mark of autophagosome, they nicely colocalize and they form these punctus, suggesting that ER-Phagy is occurring. Not only that, she also did an immunoprecipitation where in the presence of hypoxia and normoxia, and she could show that the LC3B immunoprecipitates with FAM134B. So taken together, all these suggest that there is ER-Phagy happening, and it is this specific receptor FAM134B which is involved in targeting damaged ER into autophagosomes. And this is a mouse breast cancer tissue. So she did a staining for FAM134B because those things were done in vitro. We wanted to see if a similar process also happens in vivo. So we used a mouse breast cancer model, where a PYMT model, where we could also show that similar to the in vitro, we can also see FAM134B and LC3B colocalizing. Now, so we know that the FAM134B, the ER-Phagy receptor, is involved in ER-Phagy in cancer cells during hypoxia. The question is, what is the significance of it? So what Sandhya did was she knocked down FAM. When she knocks down FAM, what you can see here is there is an increase in the ER stress response mechanisms, suggesting that there is more stress. When there is more stress, the ER stress response can be read through by studying these proteins. And as you will clearly appreciate, when you knock down FAM134B, you have increased amount of these proteins, suggesting that the ER stress increases when you knock down FAM134B. Not only that, the viability of the cells, when you knock down FAM134B, the cancer cell, you can in depth in these cancer cells. So what we could say is that depletion of this FAM134B, this increases ER stress and it reduces cell survival. Now, we wanted to dig a bit more into the mechanism as how this is happening. We have ER-Phagy, we have the ER-Phagy receptor FAM134B, we have identified that, but how is it sensing? If you look into this picture, most of the ER-Phagy receptors that have been identified, they either have this luminal domain which goes into the endoplasmic reticulum. So, and what happens is when there is misfolded proteins or proteins that are not folded properly, they accumulate in this lumen. So if they are accumulating in the lumen, then there needs to be something that goes into the lumen and senses there is accumulation of that. However, we could say that we didn't find a role for these, the other ER-Phagy receptor, but we only found for FAM134B. And as you see here, the FAM134B does not extend its arm into the lumen, which means that there is no way that it can sense something within the lumen. So that intrigued us. So how is it that FAM134B is able to sense something within the lumen? And then we thought it is probably BIP. So the BIP is an ER chaperone. So what is a chaperone? So BIP, what it does is it senses if there is misfolded proteins accumulating within the ER lumen, it senses that and it tries to unfold. So if there is the misfolding there, it tries to unfold and puts it in a proper shape here. So when BIP dissociates from the ER stress response and it goes in, senses this. So we thought probably it is BIP that goes in, senses the misfolded proteins or unfolded proteins, and comes back and talks to FAM134B. So we thought, okay, is BIP involved in this ER phagy? And as you will see here, when we knock down BIP, here, so when you induce hypoxia, as I showed before, FAM levels go down. But now when you knock down BIP, we don't see this degradation of FAM, which suggests that, probably BIP is also involved in this process of ER phagy. And consistently, what we also saw was when we knock down BIP, there is reduced cell proliferation. So then we asked, so if BIP has to go into the ER lumen, sense it, and comes and binds to FAM134B, which means that the BIP and FAM, they have to come together, they have to speak to each other. So Raja, in my lab, along with Sandhya, they did some immunoprecipitation experiments, and as you will appreciate from here, when you induce hypoxia using this COCl2, you can see that the BIP binds with FAM134B. So you immunoprecipitate FAM134B using an antibody, and you do a Western blot for FAM, for BIP, and you can see here, compared to the control, there is increased binding of FAM increased binding of BIP to FAM134B, suggesting that this BIP and FAM134B, they talk to each other. Not only that, we could also show in the mouse breast cancer tissues, as you would appreciate here, this is a larger view, and one of this has been zoomed here. As you will see in some of the regions here, the green and the red, they colocalize to give this yellow signal, which suggests that the BIP and FAM134B, they nicely colocalize with each other, which means they bind with each other. Now, it was all nice and good when until this, we have defined a mechanism as how this process is happening during hypoxia, all well and good. But then we had to start. So Sandhya, even before coming here, she had this thought that can we identify molecules which can do something with to hypoxic cells. Therefore, we prevent their proliferation. And since we had the BIP as a BIP and FAM coming together, we thought, can we find something that would target BIP? And this is what she did. She did some. I mean, if you have any doubt, these are out of my area. But nevertheless, I'll try to explain better. But if you want something more, please speak to Sandhya. She can explain it to you much better than what I do. But I try my best. So what you can see here clearly is that this is again what she said that sorry, it's marked wrongly. It's BIP. So this drug, one particular drug which we named, has the better binding affinity to BIP compared to all the other molecules. And then we collaborated with Vignesh, one of my collaborators and friend from University of Bonn, and he did some molecular dynamics of this molecule. And what we could see is that it nicely sits within the pocket of BIP, and not only that, and it specifically binds to some of the amino acids within that pocket. And he also did some molecular dynamics for us. And what we could see is it specifically binds to an arginine residue and a serine residue. And these are the molecular dynamics which were done for up to 100 nanoseconds. And you can see that the stability of the binding is pretty strong, suggesting that this is a good candidate to go ahead and work on to see that if it really inhibits BIP. And so then we went to the wet lab and we asked those questions, what does this molecule do? And clearly it prevents the accumulation of HIF1 alpha. That's the first thing that we see. And also you can see here hypoxia degrades FAM134B, and now you treat with this drug and prevents the degradation, suggesting that it clearly inhibits ER-phagy, the ER-specific autophagy, and importantly, as I showed you before, the BIP and FAM, they interact, but in the presence of hypoxia. But now when you treat with the drug, this interaction is highly reduced. And that clearly suggests that when this molecule goes and binds to BIP, it prevents the binding of FAM to BIP during hypoxia. And not only that, and you could clearly see here there is increased, this cell dies when you treat with the small molecule. And consistently we can quantify and we can say that we are able to reduce the cell viability to a highly significant extent. So I think without boring you much, I'm just coming to a conclusion now where we can say that during hypoxia, in a hypoxic cancer cell, there is accumulation of misfolded proteins. And what we have found is this misfolded proteins are sensed by the ER chaperone BIP. And once the BIP senses the ER chaperone, it goes and binds to FAM134B, and which sets the trigger for the FAM134B to target the damaged part of the endoplasmic reticulum into autophagosomes. And these autophagosomes, then they fuse with the lysosome and they are degraded. So it is a clearance degradative mechanism that removes the damaged part of the endoplasmic reticulum and thereby allowing the cells to restore ER homeostasis. So the damaged part is gone. Now the ER can grow and they can do the functions normally. So I'm really glad that we were able to do this work in collaboration with JSS, and I would like to thank the funding bodies, especially DST, which supported Sandhya's fellowship and allowed her to come here and work with me, and the European Association of Cancer Research, which also provided funds for Sandhya to come to my lab in Germany. And I'm ever grateful to my funding bodies like DFG, which funded my lab in Germany, and Center for Cancer Biology, where I'm working, and University of South Australia. And I should say that Sandhya did the 95% of the work. So it's the first time I'm presenting a work which I have done in collaboration with a person from JSS that's so dear to my heart. And Raja, who was a senior postdoc in my lab, helped Sandhya quite a bit. And I'm really indebted to Vignesh because Vignesh helped us with the molecular docking studies. I spelled it wrong. It's Vincenzo. So following upon this study, we are still taking the study further into in vivo models. So Vincenzo, who is from Uni Campania, Italy, is helping with that. And Adam Antevy is a long-time collaborator of us. And so I think this is the best that can be said. I hope you guys are not sleeping and awake to ask me some questions. So I'll end there, and I'm happy to take questions.
Thank you, Dr. Nirmal, and we must congratulate Sandhya for the excellent work what she has done in collaboration with you. Let us open the session for the question and answer. I request the participants, if you have some questions or some interaction to be done with Dr. Nirmal, the session is open, and either you can unmute yourself and ask, or you can put it in the chat box and we will ask. I request Dr. Rajesh to moderate the session.
I'm happy to see people as well. I don't know how many people were, because when I was giving the presentation, I could see only my screen and I could hardly see anything else. So I'm happy to see.
There are around 120 participants who are there with us in this presentation.
Oh, that's massive. I'm glad. And thank you all for taking time to listen to my talk. And as I put in the last slide, I hope not many of you have slept on your laptops and you're still awake.
It's a really good topic, sir, because Hello, Dr. Nirmal Robinson. What you can do is you can just stop the presentation so that you can see the participants.
I did stop the presentation now.
Yes.
So that everybody can see.
Any questions from faculty members?
Can I? Nirmal, always good to listen to you. The years go far, and then your data is getting stronger and stronger. I'm happy that you accommodated one of our students and gave her opportunity to work, and you presented excellent work. And we are glad that it had a very productive outcome, not only for her personally, but for the institution as well as the university. And so I think we really appreciate your involvement, and I think we look forward more such involvement and more opportunity for our PhD scholars as well as the postgraduate students.
I just wanted to say something here, because the reason why I thought I was contemplating between presenting some of the different work, but then I thought it would be an inspiration for other researchers from JSS as well, as not to feel bogged down thinking that you cannot do something. It is always possible to do research when you put your heart and soul into it. So that's why I thought it would be good to present Sandhya as well, so that it becomes an inspiration for many other people within JSS.
Indeed, it is, Nirmal. I have a couple of questions.
Go ahead.
My first question is, although you repeatedly said about it is a stress for cancer cells, but after your presentations, I feel that it is a favored condition for cancer cells. So, because majority of the anti-cancer as well as the non-anti-cancer drugs, they are prone for endoplasmic reticulum stress. That is one of the way that it produces some beneficial therapeutic outcome. So if that is the case, so most of the case that cancer cells take this as an advantage, or it forcefully create this environment for their cell survival or cell proliferations. What's your thoughts on that, Nirmal?
So it's a great question. I mean, what you need to think about is, like, it is an immune environment as well. So obviously we are going to be fighting against it. So tumor microenvironment, you have all the immune cells and everything. So there is a stiff competition that goes between the cancer cells and the cells from us. And you might say that it is a conducive environment for the cancer cells to grow. It is a conducive environment for a cancer cells to grow because they are cancerous. If it is not a normal cell, it is not a conducive environment for it to grow. And even if you think of the cancer cells, you would see that there is a whole lot of cancer cells which are dying there, and there is a necrotic region that is being. So what I always say is, like, if you take 10 people to Mars and put them there, probably 9 of them would die, and 1 of them would survive, or 2 of them would survive, and they would give birth. And when they give birth, it is going to be, the mass cannot hold them. So that's what it happens here as well. And even in terms of the cancer therapy as well, that's similar to that. There would be, 1 million of cells, let's say 900,000 of them, they die, but the remaining of them which survive that, and that's what is the key. And once they survive, it's massive.
Yeah. In line to the same questions, like it is interesting to see that. Do you think that each cancer cell would go through the similar environment, or the cancer cells, when they multiply, they come up with some kind of adaptive mechanisms? Those of what kind of mechanisms could be? So, like, does all the cancer cells needs to go through this ER-induced stress, or maybe some of the cells that are proliferated or replicated, they have already a pre-existing mechanism or adaptive mechanism, so that they don't have to care of hypoxia or endoplasmic stress?
So the ER stress is a common mechanism that any cell and everything that goes through, because it doesn't have to be a cancer microenvironment. It can be, an infection, or even in a wound area where there is an injury or something. Even there, you could have some kind of a stress. But the key here is that some of these cells, they use this response mechanism very effectively, and when they use it very effectively, they are. So it's not that, okay, they would either, not all of them would go through hypoxia, because the ones which are in the peripheral region, they are well, they get enough of oxygen and so on. So the ones which are in the inner regions, where there is a necrotic lesion as well, so which prevents the oxygen to reach, and you know that the angiogenesis is not proper. So the blood vessels, they don't reach into the deeper regions of the tumor. So it's not that all cancer cells would experience this, but once they experience this, they develop this kind of resistance mechanism. And once they develop increased resistance mechanism, then no matter whether, even if you treat with a drug, and the drug is going to induce the same kind of response, and the cancer cells would react in the same manner. So that's the whole thing. Once they respond to this similar stress, such as hypoxia, once they have educated themselves on how to respond to a stress like that, then they go on to respond pretty well.
This is my last question, Limal. So, having said that, this is a nice concept, but there are concepts evolving that cancer cells also promote angiogenesis. If the hypoxia is a favored condition for cancer cells, why cancer cells also promote angiogenesis?
Because, as I said, it's not, hypoxia is not a favorable condition, but the cancer cells, they develop themselves to become favorable. It's not that all cancer cells, they love to, if you put them in a normal, like when you take cancer cells and you throw them in incubators, they just enjoy growing. But when you subject them to hypoxia, they develop an additional mechanism which helps them to survive even those stringent conditions. And once they survive those stringent conditions, then, they have a whole weaponry that is ready for them to fight any other condition. And coming to angiogenesis, it is not a proper blood vessel. It is a maladaptive angiogenesis that happens. VEGF and everything, you do see blood vessels and everything, but as I said, the blood vessels are not formed properly.
Yeah, excellent. Great work. Congratulations to you and Sandhya.
Thanks.
And any questions from student side? PhD.
Praveen, good to see you.
Yeah, thank you. And this is the time for...
Sir will have...
Sir, you can do it. So this is the time for a lot of thanks. Before that, sir, really the topic which you have chosen was really interesting, because most of our scholars are working on necrosis and cell necrosis. So really this topic will be useful for our students. And not only that, even I highlighted various receptors, pathways that can make on undergo apoptosis and cell necrosis. Particularly autophagy, you have highlighted, and definitely this will be the good topics for our students, sir. I thank for that. And now this is the time for... So I start out of thanks. Respected and our most distinguished speakers of the day, and Dr. Nirmal Robinson, Adjunct Faculty of JC Sahar, Mysore, respected Principal Dr. Dhanpal, our beloved Head of the Departments, Honorable Professors. First of all, I would like to propose hearty vote of thanks for our today's speaker, Dr. Nirmal Robinson, for gracing with the good topics of today's sessions. And thank you, sir, for your very interesting and thought-provoking address. I would like to thank our beloved principal for his presence. And I would also like to thank all of our head of the departments. I'm happy to express my sincere thanks to our staff members of various departments. Finally, the wonderful students of PhD and PG students who have turned up in such big number, as Ashish Wadhwani said, not only from our department, even from other departments and the external students of other colleges. Thank you so much for your cooperations. Once again, I thank all for your cordial cooperation, sir. Thank you. Thank you very much.
Just I'd like to.
Rajesh, with the permission. Am I audible, sir?
Yes, please, sir. Go ahead, please.
Thank you, Ashish. Well, good morning. Sorry, I did not use the unmute button.